Do you want to explore immune repertoire data, discover therapy-specific clones, and create publication-ready visuals? And all this without writing a single line of code. Welcome to our guided series of immune analysis in Platforma. Over the next few videos, we'll take you through the entire workflow, from raw data to discovery. We'll cover the clonotyping analysis, the clonality of our repertoire data, clustering of clonotypes, and exploring the motifs, differential analysis to find clusters or individual T cells and B cells. Will also show how to visualize the entire dataset on an interactive clonotype space UMAP and build comprehensive visualizations or just data export to track your best lead candidates across time points or other conditions. Before you can get to those discoveries, area analysis starts with one crucial step: importing your data. And this first guide will show you exactly how to do that, covering common scenarios you'll encounter. Let's get started. First, let me create a new project. This will be a VDG project where we start our analysis from raw FASTQ files. Every project begins with the first block added automatically, samples and data. And this is the central place where you define your samples, where you bring your biological metadata about the project. And the first thing we see here is bringing the raw files into the project. And this can be done in two ways, either by importing the files that are local on your personal computer, or that's the case for our guide, using the remote storage if our backend is connected to the data storage with the raw data. So let's select some files, and in the first example, I'm going to use the most frequent data type. It's just paired FASTQ files. So I selected all the FASTQ files, and as you see here, the platform automatically recognized the sample name from the file name, read1, read2 structure, etc. If we have, for example, lanes, then lane information will be also inferred. We can modify the pattern, and that's actually I will show you later, to fine-tune the way how we infer the information from the file names. So let's assign a meaningful name to this data set. So this is bulk VDG data, and create the data set. Now we have our samples, and for every sample we have a pair of FASTQ files. So we have our first data set. And actually, in this project, we have multiple data sets. This is the bulk VDG data, but in this project, we also have single cell 10x genomics gene expression data set, and let's add it. That's actually what you should do. You import all of the data sets that you have in the project. So let's select all of our FASTQ files for 10x genomics gene expression data here. And you see now we have another data set, and the structure of the file names different. So we have same sample, we have lanes, and actually for every sample we have 4 lanes here. We're ignoring index files because we don't need them for the analysis. And let's call this data set 10x GEX. This is gene expression data. And you can note one thing here: S1, S1, S10, S10, etc. So, well, the way how those samples are named contains redundancy. So let's fix it by modifying the pattern and to get rid of the second S1, S2, etc. So, and that can be done easily by modifying the pattern and excluding those piece of the file name as from the sample. And that's it. Let's create our sample, our data set. Now we see for every sample, we have multiple lanes. For every lane, we have a pair of FASTQ files. So now we have 2 data sets, and the next crucial step in any project is actually to describe the meaning of those samples for downstream analysis. I switch to metadata section, and here there's an import metadata option allowing you to pick Excel or TSV or CSV file with metadata information about your samples. The only requirement for the Excel file is to have a column which contains sample names. So I clicked import and imported various metadata columns. They are absolutely arbitrary. You may have whatever metadata you have in your Excel file and match those metadata columns with our samples. So for example, in this specific study, we have a time point, the most important metadata column. So that's it. Now we click run to make import actually happening. It's pretty fast, and we have our project defined. Now we are ready to proceed with the next analysis steps by adding the dedicated blocks. In this specific project, for example, to analyze the bulk data into clonotypes, we should add MiXCR Clonotyping block, which takes the bulk data set as input, where we can specify the parameters of the analysis by just picking the name of the chemistry and clicking run button. We'll have another dedicated guide how to do the clonotyping analysis. I'm just showing you as an example. Or to process our gene expression data, we may choose the cell ranger block to run the gene expression analysis on single cell data with cell ranger. So, but what if we already have pre-processed files? Here, we started with FASTQ files. But what if we already have a pre-processed clonotypes as a TSV files from some other pipelines? That's also possible. And actually, let's do this in a separate project. Let's create a project VDG from XSV, and we start with the same samples and data block. And now we, in the same way, need to import the raw files, the data files, preprocessed files with our clonotypes into the project. I have some data from Adaptive Biotechnologies immunoSeq tool. I'm going to use those TSV files downloaded from the immunoSeq portal, and absolutely same way. So we have the same interface here allowing to import TSV files. Let's call this data set bulk TCR because this is TCR data, and create. That's it, easy peasy. But if we look on our samples, we see that actually that's not the right way. Here you see that we have one sample actually and two immunological chains. Here, for example, alpha, delta, and here gamma, which obviously two files, two parts of one sample. And we don't want to consider different homological chains like TCR alpha, beta, and TCR gamma as two different samples, those two parts of one sample. So we need to modify the way how we import this data to actually take this into account. So let me remove all those samples and let me remove this data set and start over by creating a new data set and repeat this. So I am again selecting all of the TSV files that we have. And now let's modify the pattern here to match this suffix as actually the chain. And that's possible by adding the chain tag in our name pattern. So with this chain tag, this suffix is recognized as the chain. Well, first of all, what we can observe here is that for some samples, there is no matching between the pattern and the file name. Well, c'est la vie, it's clear that this sample or this sample follow a different naming convention. So this sample does not have any suffix and this sample does not have any suffix. So for some reason, well, those who created those files named them differently. And there is no other option except going manually to those files and renaming them. And this is our general recommendation. Use consistent file naming convention across all of your samples. If you use an underscore, use it in all sample names. If you use dash, use dash in all samples. When you mix them, well, it's still possible, but not recommended. So let's call this dataset bulk TCR and create it. In the same way, we can add metadata, but for the purpose of this demonstration, let's just click run. Well, first of all, you see that here we have one same sample, and for each sample, we have multiple chains. So we did this job. And in the metadata, we have every sample only once, listed only once. So the next step in this case is to actually import the clonotyping information from those TSV files, which we can do with import VDJ data block. This is a dedicated tool allowing to import the clonal information from the TSV or CSV files from preprocessors. So here we again select the dataset, bulk TCR, and the data format. We may choose the custom format and provide the mapping between the columns and the files and the information that we need to define the clonotype. But since we use adaptive biotechnologies immunoseq here, we're just picking up immunoseq, and that's it. Now we can click run, wait to the import of the clonal information be completed, and that's it, done. So once we did this, either with importing VDJ data or with analyzing it with MiXCR Clonotyping, we are ready for downstream analysis that we will cover in the following videos.